Hi and welcome to our last macromolecule. We are finally approaching nucleic acids. Nucleic acids are near and dear to my heart because I am a molecular biologist by training. So when we talk about nucleic acids, we're talking about DNA, we're talking about RNA, and ultimately this is the code within every single nucleus and every single cell in your body that gives you those special traits and characteristics that we can see, and how all this functional stuff inside your cells that we can't see, how they all work. And so DNA is something beautiful and amazing to me. And so we're just going to get a glimpse into the structure and function of nucleic acids. We're going to really get into DNA and RNA later when we get into our molecular biology unit later. So I'm not going to go into too much detail now, but I do want you to be familiar with what DNA is and what RNA is. So this is our last unit here, and I want to say we're on Roman numeral 7, which is nucleic acids. So nucleic acids. So whenever you hear the word nucleic acid, I want you to automatically think DNA or RNA, because that's what they are. It's just DNA or RNA. But what nucleic acids are, the broad definition is that these are long polymers made of nucleotide monomers. So remember, every polymer we've learned about has its own special monomer out there. In the DNA and RNA world, the monomers are called nucleotides. So there's a lot of vocabulary I'm throwing at you in this unit, but I promise if you just keep using it and studying it, it's going to become second nature too, so it's going to be good. So nucleic acids essentially are DNA and RNA. We're going to define those guys later. DNA and RNA, and these are long polymers, and we call these polymers polynucleotides. Made of nucleotide monomers. So let's talk about what a nucleotide is. So a nucleotide, which is the monomer for nucleic acid polymers. So nucleotides, they're made of 3 different parts, if you will. So composed of 3 parts. So the first part is what we call a nitrogenous base. Now, what the nitrogenous base is, it is basic in nature, that's why we call it a nitrogenous base, but it has a lot of nitrogen in it as well. But there are four different kinds of, actually technically 5, but in DNA there's 4 different kinds of bases out there. And if you've ever watched, enough, bio documentaries or anything about DNA or anything like that, you know that there's 4 letters that make up your DNA. The bases, those are the letters they're talking about. So there's cytosine, which is C, guanine, which is G, adenine, which is A, and thymine, which is T. So ACGT, that's kind of the alphabet, if you will, for your DNA. However, in RNA doesn't have T's, it only has U's, which is uracil. So what you need to know is that RNA basically has no T, but it has uracil, which is U instead. I'm not going to draw the structure of these bases. They're pretty complex. Once you do get more into the molecular biology world, you will see how beautiful these structures go together and how they pair up together. We're going to talk about that in a moment, though. So the nitrogenous bases, that's your alphabet. It's your G, C, A, T's, all that stuff. That's one part. So you could have one of those in your nucleotide. The other part of your nucleotide is a 5-carbon sugar. Now, depending if you're DNA or if you're RNA, the sugar is going to look a little bit different. In DNA, the sugar is called deoxyribose. The os, remember, that's a sugar thing. In RNA, it's just ribose. So DNA has deoxyribose. RNA is ribose. And that's where we get the name for DNA and RNA. DNA stands for deoxyribonucleic acid. RNA stands for ribonucleic acid. So the sugar is what determines if you have RNA or DNA, along with a few other properties, but that's like the main, the backbone, if you will, that can tell you if you have DNA or RNA on hand. Deoxyribose and ribose look very similar. As the name suggests, for DNA, it's deoxyribose, meaning the difference between ribose and deoxyribose is literally 1 oxygen. So DNA has 1 oxygen less in its sugar than RNA does. That's the only difference between the two. But again, 1 atom makes a big difference on structure and function. DNA is not RNA and RNA is not DNA. They both have their own separate but equal functions in the cell. So it's really amazing what 1 atom can do. And lastly, the last part of your nucleotide is a phosphate group. Good old PO4, just the same functional group we've always had. So if you look at the structure of a nucleotide, the shorthand way to draw these is you draw your 5-carbon sugar, and it's a pentagon with an oxygen at the top, and it has a carbon up here. But coming off this carbon, you have your phosphate group right there. So you have your phosphate group coming off the carbon, and then on this carbon right here, you have your base. The even easier way to draw this, because this takes time and people don't want to do that, you would do this instead. You would draw base, and you could, in this base, you would either put an A, T, G, C, or U, if you're talking about RNA, in there, and then you would draw your phosphate. That circled with the P on there, that's the phosphate symbol in DNA land, okay? So that is what your typical nucleotide looks like. You got your nitrogenous base, you got your 5-carbon sugar, and you have your phosphate group. And that is what's going on here. Now remember how we number things in organic chemistry when we're talking about carbons? So you start at the oxygen and you work your way around clockwise. This will be carbon number 1, carbon 2, 3, 4, and 5. This carbon is that carbon right there. So 1, 2, 3, 4, 5. There's your 5-carbon sugar. Remember this numbering, it's going to come back in a minute. So that is how you create a nucleotide. What I want you to know really quickly are the differences between DNA and RNA. We're not going to go into their functions yet, we will later, but let's talk about really quickly the differences between DNA and RNA. So this will be big B under your notes. So when you're talking about DNA, everyone knows about DNA. For the longest time, people didn't realize how important RNA was. And now we're realizing more and more RNA does a lot of good stuff in the cell. So, but we're not going to talk about those awesome differences yet. So let's talk about the basic differences. Differences between DNA and RNA. Okay, switch pens here, get a little darker. Differences between DNA and RNA. The first major difference, if you were just to look at DNA and RNA, you would see a big major difference. DNA is this classic famous structure of a double helix. It's essentially a polynucleotide chain, 2 polynucleotide chains helicizing together, and they make this gorgeous alpha helix, very much like a twist, not a twisted ladder, but like a like a staircase ladder in a way. It's beautiful. So DNA is a double helix made of 2 polynucleotide chains. So if DNA is a double helix, you could think RNA can't be. So RNA is actually a single helix made of 1 polynucleotide chain. RNA is a single helix. Okay, the last 2 differences we've already talked about, just to reiterate them. For the bases, DNA uses the bases A, T, G, or C. So DNA nitrogenous bases are A, T, G, or C. And RNA nitrogenous bases are A, U, G, or C. And the last difference we've already talked about as well, but so DNA, 5-carbon sugar is deoxyribose, and RNA, 5-carbon sugar is ribose. Okay, now that we got the differences out of the way, now we could just talk about building a polynucleotide chain. So polynucleotide chain is exactly what it sounds like. It's taking all these nucleotide monomers and linking them up together. So let's make a polynucleotide chain, and then you can see how RNA is made, and if we put 2 of these chains together, how do you make DNA? So here we go, big letter C, constructing a polynucleotide chain. Okay, so these are a little bit different. Again, once again, you're going to be doing dehydration reactions and all of that, but I'm not going to show you the functional groups on how they interact to do this. We're just going to show you how the chain works, because I want to get you to the point where you can start to see how the double helix looks. So let's take a nucleotide. We'll just do a basic structure here. You got your base, and you got your phosphate group over here. We're keeping it very simple. I'm not showing you all the functional groups and how they interact this time around. But what I do want you to know is the numbering here. So I have 1, 2, 3, 4, 5. The way our DNA links together is this nucleotide is going to link together with another nucleotide, but here's how they interact. This 3rd carbon has a hydroxyl group coming off of it. It's actually going to interact with the phosphate group of another nucleotide. Okay? And then once again, we do another phosphate group. So here is my polynucleotide chain made of 3 nucleotides right there. This link between each nucleotide, this link right here, that is called a phosphodiester bond. You've got to know that. So this is called a phosphodiester bond. All right, so phospho because there's a phosphate, diester because there's 2 esters being involved in there, and you got your bond. So phosphodiester bond. So esters come in handy for linking things together, just like they did in the lipid world. We see it again in the DNA world. But this time we're making a true polymer. You could keep going down, 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 and you would see how long your DNA can get. You have oh gosh, billions of DNA linked together, nucleotides linked together in your cells as we speak. In every single cell, you got about, oh gosh, 3 feet of DNA that you package up in there. It's pretty amazing. So that is what one polynucleotide chain would look like. So this whole structure, this whole thing, this would be a polynucleotide chain. And the other thing I want you to realize is the order of how the polynucleotide chain is going. If you're counting the carbons, is what we have to do in the DNA world. I have my number 5 carbon facing up in this direction, and my number 3 carbon, the closest one to the bottom, in that direction. So the way we order this, we actually say this polynucleotide, this would be the 5' end, and this would be the 3' end. Prime is just giving you the direction on which one's facing closest to what end. So 5' is here, 3' is here. So this would be your typical RNA. So let's make DNA, because DNA is a double helix. We need to put 2 polynucleotides together. So in order to put 2 polynucleotides together, I just can't stick these 2 together. What's going to happen is I actually have to flip this over and put it together, because the way they connect, the bases connect together on the inside. Each base is specific for a different base. And here's what I mean by that. They complement each other. So if this base right here was an A, the only other base that can go right here to make this binding, this connection work, has to be a T. And vice versa. If this was a T, that had to be an A. So here's how it works, and I'm gonna beg your patience as I draw this, because I have to go upside down. And do you notice how the structure for my DNA is actually flipped? And so let's continue this. Let's make up some more bases here, and I'll show you what's going on here. So if this was a G, G's can only connect, complementary bind, with C's. C's can only go with G's, G's can only go with C's. These are your 4 nucleotides. A's and T's always go together, G's and C's always go together. And so here is my next. It's going to be a longer bond right there. There's my polynucleotide, or my phosphodiester bond right there. So let's make up another base right here. We'll put a T here this time, and we'll make this one. If this is a T, this has to be A. So okay. It's tricky drawing these, but here's what I want you to see from this. Notice how on this side of the polynucleotide chain, my carbon on my 5th carbon is facing up here, and my 3rd carbon is the closest one down to the bottom. You're going to see that exact opposite. It's anti-parallel, we call it, on the other side. So if I were to count these carbons, this would be 1, 2, 3, 4, 5, and I'll go down to the last one. 1, 2, 3, 4, 5. My 3rd carbon is the closest one to the top, whereas my 5th carbon is the closest one to the top on this side. And now my 5 carbon is on the bottom, whereas my 3 is on the bottom here. So they're anti-parallel. That's what gives DNA its lovely double helix shape. They have to be anti-parallel. That's the only way they go together. There's been so many experiments, and then back in the day, Rosalind Franklin, Watson, and Crick, they all figured out what the structure was of DNA, and we'll talk about that more later. So this is how you construct a polynucleotide chain. This is one chain, and this is the second chain. You put them together, you have a double helix. The way these bases connect together is with hydrogen bonds. So you have hydrogen bonds between your A's and your T's and your G's and your C's. So these guys right here, these are hydrogen bonds. Now if you notice, I only put 2 hydrogen bonds between A and T and 3 between G and C, because that's actually how many hydrogen bonds there are between those bases. G's and C's have 3, A's and T's have 2. So the hydrogen bonds are actually very weak bonds. But when you have many of them together, it's very strong. I want you to think of Velcro. Like one little thing of Velcro is super easy to break. Put a whole bunch of Velcro together, you got to put a little energy into it to break it apart. That's how hydrogen bonds work. So your DNA isn't going to readily just unzip. You actually have to apply heat and some energy into breaking that apart, and it doesn't happen very easily. But the beautiful thing is, if you do heat up DNA, say to boiling, like 99, 100 degrees Celsius, your DNA will unzip. And if you cool the water back down, your DNA will go right back together again, exactly where it needs to be. It's awesome. So that is how your DNA works. You have phosphodiester bonds between nucleotides, hydrogen bonds between bases on a double helix on DNA. So let's write this stuff out really quick, and then we'll be done with our DNA. So constructing a polynucleotide chain, this will be number 1 under that, under this whole picture. You need to know that phosphodiester bonds connect nucleotides on a polynucleotide chain. Hydrogen bonds connect bases between 2 polynucleotide chains. And lastly, in order for 2 polynucleotide chains to be together in DNA, the polynucleotide chains are anti-parallel, meaning you have a 5' to 3' direction on one side and a 3' to 5' direction on the other side. So lastly, in DNA, polynucleotide chains are anti-parallel. Meaning you have 5' to 3' on one side and then 3' to 5' on the other side. And that's it for nucleic acids. So remember, nucleic acids, you got the monomer of a nucleotide made of a nitrogenous base, 5-carbon sugar, phosphate group. They link together via phosphodiester bonds to create a polynucleotide chain, which has a certain direction, 5' to 3', and then another polynucleotide chain comes and links together with it via hydrogen bonds between the bases, and that will have to be anti-parallel, so in this case it's 3' to 5', and that's how DNA looks. RNA would just be one side of this. So thank you for sticking through all of those macromolecules. I'm going to do that review sheet with you next. So if you haven't already done so, go ahead and download. It's a table with all the different macromolecules, and we're just going to fill in that table if you want to give it a stab at it yourself and then come back and check with me. I would love that. And then that'll be a great study guide before you go and take your test on it. So thank you for sticking through probably one of the longer sections we have, but it's foundational, and then we're going to keep moving forward. So see you soon.